Hello my dear children, we are studying unit 2 of advanced level biology syllabus, the competency level 2.1 chemical and cellular basis of biology. We have already studied about the importance of water in life and the major organic components. We have finished about the carbohydrates. Now I hope to discuss rest of the organic components in living matter. First is the lipids. Now you know lipids are water insoluble. That's why we say it is a hydrophobic molecules, hydrophobic. Insoluble in water, some dissolve in organic solvents. They are large biological molecules, but they are not polymers or macromolecules. Lipids consist of carbon, hydrogen, oxygen as in carbohydrates. But you know in carbohydrates, hydrogen and oxygen ratio is 2:1. But here the ratio is not 2:1, more hydrogen in these lipids. So there are different types of lipids in nature, but in our syllabus there are 3 major types. They are the fats, phospholipids and steroids. First we take the fat. Fats are made of glycerol molecule and 3 fatty acids. 3 fatty acids. So glycerol is a type of organic alcohol. It has 3 hydroxyl groups. 3 hydroxyl group. I showed you the 3 hydroxyl groups in the glycerol molecule. And fatty acids are with long hydrocarbon chains, about 16 to 18 carbons in their chains. So at the terminal end it has a carboxylic acid group. It has a carboxylic acid group. So this is the structure of the fatty acids and glycerol. Now let us see how this fat molecule is formed from this glycerol and fatty acids. So one glycerol molecule combined with the 3 fatty acids molecule. This is also a condensation reaction. Now let us see carboxyl group of fatty acid molecules bind to each hydroxyl group of glycerol. Now look at that. Carboxyl groups of 3 fatty acids molecules bind to each hydroxyl groups of glycerol and remove as 3 water molecules and form the fat molecule. It is named as triacyl glycerol. Triacyl glycerol. Now you can see the 3 water molecules removed here, and this is the ester bond formed between this glycerol and 3 fatty acids. So this is the fat molecule, fat or oil. Then now hydrocarbon chains of this fat, hydrocarbon chains of these fatty acids actually contribute to the hydrophobic nature of the fat molecule. I told you all lipids are hydrophobic. So hydrophobic nature is given by these hydrocarbon chains of the fatty acids. Based on the nature of hydrocarbon chains of fatty acids, I think you have heard about this saturated fats and unsaturated fats. So saturated fat made up of saturated fatty acids, hydrocarbon chains of these fatty acids having no double bonds. That means only single bonds are present in between carbon-carbon atoms. But unsaturated fatty acids, they are also made up of long hydrocarbon chains, but in this chain they have 1 or more double bonds. one or more double bonds. So, these are unsaturated fats. Now, I will show you these two types of, that is saturated, only single bonds are there. Unsaturated fatty acids, you can see the one double bond here, one double bond here. Then let us see the differences between saturated fats and the unsaturated fat. So, saturated fats made up of saturated fatty acids, unsaturated fats made up of unsaturated fatty acids. And again, hydrocarbon chains lack any double bonds in saturated fats, only single bonds. But unsaturated fats, they have one or more double bonds in their hydrocarbon chains. And again, saturated fats are usually animal fats, but unsaturated usually plant fats. So, examples given are, now here butter, they are actually mostly solid at room temperature, but unsaturated mostly liquid at room temperature, especially types of vegetable oil. But remember, some foods products such as coconut oil, palm oil or whole milk remain as liquids at room temperature. You know, they are liquids at room temperature, but are high in saturated fat. Then in unsaturated fat, again there are two types. They are cis unsaturated fats and the trans unsaturated fats. The two categories based on their double bond. Now, you know, unsaturated fats, they have the one or more double bonds in their hydrocarbon chain. So, based on their double bonds or the nature of the double bond, there are two, cis and trans. Now, these are the example given. Now, here cis fat molecule and here trans fat molecule. Now, you can see the hydrogen atom. Now, here this way and here down side. So, trans fat. Now, composition of excess saturated fats and trans and unsaturated fat contribute to increase LDL. LDL means low density lipoproteins. That means in high cholesterol. Now, in a lipid profile test done by you, it says the amount of HDL, LDL and the amount of triglyceride. Now, low density lipoproteins are high with bad cholesterol. High density lipoproteins are rich with good cholesterol. So, if you take saturated fat and trans unsaturated fat, it is not healthy for you. So, the trans fat can be made from vegetable oil through a process called hydrogenation. Now, this is actually types of saturated, unsaturated, cis and trans. Now, look at this saturated, they have only the single bonds between carbon-carbon atoms. Here the unsaturated linoleic acid, it has a cis bond. So, this is good type of fat found in vegetable oil, but here trans. Now, from this cis unsaturated fat, trans unsaturated fat can be obtained by hydrogenation. So, here now you can see the bond is somewhat changed here, but anyway this is also linoleic acid, but it is transformed. Hydrogenated fats. Polyunsaturated vegetable oils. Poly means, now you know there are two types of unsaturated, because mono unsaturated and polyunsaturated. Mono means only one double bond in their hydrocarbon chain. Poly means there are several double bonds in their hydrocarbon chain. So, polyunsaturated vegetable oils to which hydrogen has been added commercially to make them solid at room temperature. You have seen some margarine. Now, some margarine is made of sunflower oil and then the canola oil, like. So, they are margarine. So, these are hydrogenated fats. So, this process called hydrogenation. That means the turns polyunsaturated vegetable oils into saturated fats. So, that is actually not good for the health. So, margarine and shortening are made in this way. What are these? Now, you know margarine, shortening are now these are used to make some bakery products. So, that is also not good for health. The hydrogenated fats are linked with diabetes, heart diseases and obesity. Now, I will show you what are the good fat and what are bad fats. Now, you know trans fat and the saturated fats are not good, but unsaturated fat, this actually with cis form, they are good for health. Now, look at that the example. Now, trans fats are actually hydrogenated vegetable oil, that means they are actually unsaturated, but after hydrogenation then they become the bad fat. Now, example given are fast foods, cakes and then pastries, chocolate, deep fried food. And saturated vegetable fats, that is coconut oil, palm oil and some beverages, creamer, condensed milk. Animal fat, poultry skin, fatty meat, butter, ghee, lard, full cream, dairy products, full cream milk or cheese also. And these are the unsaturated, some are polyunsaturated, some are monounsaturated. Now, look at this, especially all nuts, walnut and the peanuts, right, all are monounsaturated. Again, this polyunsaturated, corn oil, soya bean, sunflower oil, seeds, cold water fish, salmon. Cold water fish means they actually live in cold areas of the ocean. So, they are good for, or they are polyunsaturated. Then let us see next type of lipid, phospholipids. Phospholipids are major components of the cell membrane, you know about that. They are composed of two fatty acids and one phosphate group attached to the glycerol molecule. Now, in fact, 3 fatty acids are attached to the glycerol molecule. Here only 2 fatty acids and 1 phosphate group. So, the phosphate group gives the negative electrical charge to the molecule, and typically an additional polar molecule or sometimes small charge molecule is also linked to the phosphate group. So, example given is choline. And the other interesting feature in this phospholipid molecule, because the two ends of phospholipid show different behavior. That means the hydrocarbon chains of the phospholipid are hydrophobic. And then the phosphate group and the other attached molecule, choline or some other molecule, gives hydrophilic nature. Hydrophilic, that means they like water, but hydrophobic means they do not like water. Now, this is the structure of phospholipid molecule. Now, look at that, the yellow colored chains are the hydrocarbon chains of 2 fatty acids. There are only 2 fatty acids chains here, and then the glycerol molecule, that one phosphate group is attached to the glycerol molecule, phosphate group and the other choline group. So, this is the head of the phospholipid molecule and these are the tails. So, the head is hydrophilic and the tails are hydrophobic. And the other one means the third type of lipids, steroids. Steroids are differ from others because they lack fatty acids. but these are very important in living organism because steroids are characterized by having a carbon skeleton. Now, you can see there are 4 carbon rings here and no fatty acids. Carbon skeleton with 4 fused rings. Example are all sex hormones like estrogen, progesterone and testosterone, and also cholesterol and some other hormones also, aldosterone also, steroids. Now, this is the structure of the cholesterol. Now, you can see 4 carbon rings fused together. Now, cholesterol, you know about cholesterol. Now, I told you that bad cholesterol and a good cholesterol. Now, the bad is LDL, that is the low density lipoproteins, and the good cholesterol is HDL, high density lipoproteins. Bad cholesterol, they are accumulated with some white blood cells and they narrow the blood vessels, lumen of the blood vessels. It sometimes creates blood clots. So, that is why we say they are bad cholesterol. So, but HDL is good cholesterol. HDL is present in especially fish oil, and the unsaturated fats. Then functions of lipid in nature. Now, fat and oil we have discussed already. They normally act as energy source in all living organism because it can store energy. And then the phospholipid, that is a major component of plasma membrane. And steroids, they are the sex hormones, cholesterol. Cholesterol is also present in cell membrane, especially in animal cell membrane, and other sex hormones. And other than those, some other, I would like to inform this because this waxes, hormones and terpenes, they are also fats. Now, waxes, bee wax, and then there are in insects they have wax on their exoskeleton, and then on cuticle of the leaves it prevent evaporation of water. And then hormones. So, hormones, special signaling molecule, I mentioned those hormones, mainly sex hormones and the other adrenal cortex hormones. And then terpenes, these are very important because scents of flowers and some essential oil like menthol and then the cinnamon oil, right. So, these actually the scents given by these terpenes. And then carotenoids, chlorophyll pigments, they are also terpenes. Natural rubber contain terpenes. So, very commercially these terpenes are very important. Then how do you identify fat in the laboratory? In your laboratories there is a solution called Sudan 3. It is red in color. So, you take a food solution into a test tube and then add few drops, add about 20 drops of Sudan 3 and shake well, then leave it to settle. Then you can see the fat layer. Now, the solution contains the water and fat, so fat layer become red in color. And then you can see the fat globules also. The third type of organic molecule in living matter, that is the proteins. Proteins are macromolecules. All proteins are macromolecules. That means biopolymers. They are polymers of amino acids. Amino acids connected by peptide bonds, the long chains of amino acids. So, major elemental composition is carbon, hydrogen, oxygen, nitrogen and sulfur. There are 5 elements in proteins. So, proteins are made up of various combinations of 20 different amino acids. Now, there are lots of amino acids, but in living organisms only 20 amino acids, 20 different amino acids are involved in protein synthesis. What is this amino acids? So, this is the structure of amino acids. Now, it has a central carbon. It is asymmetric. Asymmetric means the 4 sides has 4 different groups. Now, each amino acid is composed of amino group, NH2 group and the carboxyl group, COOH group and hydrogen atom, and R means alkyl group, alkyl group. So, alkyl group is the only group that can be varied in different types of amino acids, right. So, the R group is called side chain because it is varied according to the type of amino acids. Side chain differs with each amino acid, whereas the other groups are called the backbone. So, the other groups of the molecule, do not change their structure. So, this is backbone, these 3 groups are backbone, that is amino group, carboxyl group and the hydrogen atom. But R is the side chain, it can be varied. So, this is very interesting nature of proteins. Now, this actually gives by these amino acids amphoteric nature of amino acids, amphoteric nature. So, this you know that NH group and the carboxylic acid group. So, NH group, that is amino group, has alkaline nature and carboxyl group has acidic nature, that means that it can donate hydrogen ions. So, when both characteristics are found in one molecule, both characteristics are found in one molecule, so that is why we say amino acids are amphoteric molecules. So, these molecules can react with both acids and bases. Now, look at this, the carboxyl group. Now, you know what is the carboxyl group. This is the carboxyl group. So, it can donate hydrogen ions in alkaline medium and become negatively charged, and it can combine with the hydroxyl groups in the medium and form water. Amino group can receive hydrogen ions. Now, picks up hydrogen ions in acidic medium. You know acidic medium is normally high hydrogen ion concentration. So, this amino group can fix these hydrogen ions to the NH2 and become NH3 plus. So, no change of pH of the medium. So, that is very important in nature. Now, thus amino acids and protein act as buffers in stabilizing the pH of their environment. Now, consider our some transport proteins in our blood. Now, if we have acidic food or alkaline food, but the blood does not become acidic or alkaline because of this nature, So, this is very interesting and the very important property of these proteins. These are the 20 different amino acids. I will just show you, you do not need to memorize this. These are the 20 different amino acids which are involved in protein synthesis in a living cell. Now, you can see the blue colors. Now, you can see the blue rectangles in all 3 types. These are the backbones. The yellow colors groups, you can see that they are changing different types. Some have the carbon rings also, some have sulfur in their chain. Here the methionine, they have the sulfur in their chain. right. So, different types of elements are there, and then this is the side chain. So, these are the 20 different, some are acidic in nature, some are alkaline in nature, some are hydrophilic likewise. So, I told you that proteins are polymers of amino acids, that means they are monoaminities amino acids. So, two amino acids undergo condensation reaction like in two monosaccharides and produce dipeptide by removing a water molecule. The bond formed between two amino acids is called peptide bond. Okay. Now, let us see how it forms. Now, this is the two amino acids. Now, you can see COOH group of one amino acid and hydrogen ion of the other one, that means the hydrogen ion of the NH2 group of the other amino acid. Now, you can see the OH group from the carboxylic acid group of the one amino acid and hydrogen ion of the NH2 group of the other amino acid combine and remove as a water molecule and form bond here, that is the peptide bond. So, this is actually, this molecule is dipeptides, right, dipeptide. So, large number of amino acids combine together by peptide bonds and form the large molecules of proteins. Now, proteins are different. Proteins are different types. Now, in plant proteins, animal proteins, in plants also different types of plant proteins, in animals also different types of animal protein. The nature of the protein varied based on the sequence of amino acids, because you know side chains are different, no? Side chains are differs. So, therefore, sequence of amino acids in different types of proteins. Now, there are 4 levels of protein structure which play actually when they participate in reaction, they play very important role in their function. Now, first is the primary structure. Primary structure is the unique sequence of linearly arranged amino acids. Linearly arranged amino acids, they have only the peptide bonds. Now, these are the, now blue circles are amino acids. So, in between these amino acids there are peptide bonds. So, this is linearly arranged amino acid. And then secondary structure, there are 2 types. The primary structure of the single polypeptide chain. Now, only one polypeptide chain here. Coils and folds form these 2 types of structures. Now, this is called beta pleated sheet, but you can see 2 polypeptide chain, but this is only one chain, only one single chain. Here also single chain. Now, this is alpha helix structure. Now, how they combine? Now, look at this. Now, hydrogen bonds are formed between the oxygen atom and the hydrogen atom attached to the nitrogen atoms of the same polypeptide chain. Now, look at this. These are the hydrogen bonds formed between these things. So, because of this formation, they folds and coil further. Now, beta pleated sheet can be found in spider silk fiber, and alpha helix structure can be found in keratin. You know what is keratin. Then the tertiary, more complex. The secondary polypeptide chain or the secondary structure of a protein bends, that means further bends and further folds extensively, forming a precise, compact, unique, functional and 3-dimensional globular shape. Now, look at that there are several features there. Compact, precise compact, unique functional and 3-dimensional globular shape. So, these are globular proteins. Many enzymes are globular proteins. Now, look at this. The structure is maintained by, now you know in primary structure you have only the peptide bonds between amino acids. Secondary structure other than peptide bonds, then it has hydrogen bonds. Now, look at this. This tertiary structure is maintained by the hydrogen bonds, ionic bonds, disulfide bonds, and then the van der Waals interaction, hydrophobic interaction between side chains of amino acids in polypeptide chains. Now, look at all these bond types are shown here. This is hydrogen bond, hydrophobic interaction, and this is the disulfide bond, and then the ionic bonds. So, because of this bonding pattern, they become more compact and gives 3D shape, that means 3-dimensional globular shape. So, examples are, I told you that many enzymes are in this structure, and then the myoglobin, you know, it is a blood pigment or blood protein in muscles, and then the albumin. So, these are the example for tertiary structure. Now, the final level is the quaternary structure. Now, this is differ from others because aggregation of 2 or more polypeptide chain. Now, up to tertiary structure only one polypeptide chain, but here 2 or more polypeptide chains involved in the formation of this functional protein. Now, now you can see the hemoglobin molecule. This is an example for quaternary structure. Hemoglobin molecule consists of 4 polypeptide chains. Now, 2 are named as beta chains and other 2 are alpha chains. Now, these chains are called subunits, and then which were held together by inter and intramolecular interactions. Now, other than those in this tertiary structure, some other interactions also involve to form or stabilize this structure. And you know, hemoglobin, it has the 4 heme groups, and each heme groups contain iron, metal ion, and with this 4 polypeptide chains. So, this structure is specific to this. If this structure is lost, then they can't perform their biological function. Enzymes also like that. The structure will be destroyed due to some factors, physical factors, sometimes high temperature likewise. Denaturation of proteins. Denaturation of proteins is the loss of specific chemical 3-dimensional shape due to the alteration of weak chemical bonds and interaction. Now, we discuss what are the types of bonds, right? Loss of specific, now these bonds actually help to maintain their specific structure or specific shape. But loss of specific chemical 3-dimensional shape due to the alteration of weak chemical bonds and interaction, then they can't perform their biological function. So, peptide bonds of the polypeptide chain are unaffected during denaturation. Now, remember that one. Other types of bonds, that is I told you ionic bonds, van der Waals interactions, and then the other bonds between alkyl groups. So, these will be destroyed, but peptide bonds are not affected. Now, what are the factors which destroy this structure of the protein, or what are the factors affecting the denaturation? So, these are the high temperature, very high temperature, high energy radiation. What are these high energy radiation? Examples are UV radiation and then the other IR radiation, they are high energy, disrupt hydrogen bonds and hydrophobic interaction in the molecule. And then the strong acids and the alkaline, that means strong acids and strong bases, and high concentration of salts. So, because of this they disrupt hydrogen bonds. And then the heavy metals, you know what are heavy metals? Disrupt disulfide bonds. And organic solvents and detergents disrupt hydrophobic interactions between R means alkyl groups. So, organic solvents may be benzene and then the chloroform, like that. So, factors, these are the factors which denature the proteins. But remember that the linear polypeptide chain is there. Now, look at this. Now, normal protein has a specific shape, but during denaturation then they lost the biological activity. So, they have the polypeptide chain with peptide bonds, but without other bonds. Now, you know egg, chick egg. Now, it has albumin protein. Now, what is the function of this? Actually, eggs are not for our consumption. Albumin actually provide nourishment to the developing embryo of chick. So, it is normally appears semi-solid structure, semi-solid liquid. But when we boil egg, what happen? That whole thing become solid. That means the albumin protein become white in color and solid. Now that boiled egg can't provide nourishment to the developing embryo, right? So, this protein has the polypeptide chain, but they can't perform that specific function. So, this table gives the functions of protein. There are so many proteins in nature. Now, look at this. Catalytic proteins, they are the enzymes. There are several types of enzymes. They perform catalytic action in our biochemical reactions in our body. So, structural proteins, examples are keratin and collagen. Keratin prevent desiccation. Now, we have a keratinized cells on our epidermis. So, it prevent desiccation. And then the collagen fibers are in connective tissues. So, mainly in bones, cartilage, and so therefore, it provides strength and support. Then storage proteins, ovalbumin in chick egg, and the casein, storage protein in milk. Then the transport protein, mainly hemoglobin, transport oxygen and carbon dioxide in our blood. Serum albumin, there are two types of albumin, ovalbumin, serum albumin. Serum albumin transport fatty acids in our blood. Then the hormones. Hormones are different. Some are steroids, some are peptides, and some are proteins. So, insulin and glucagon, it regulates blood glucose level. You know about this. Insulin decrease the blood glucose level, glucagon increase the blood glucose level. And then the contractile protein, that means we need these proteins for our movements, especially actin and myosin. So, they are in our muscle cells, contraction of muscle fibers. Then the defensive, they are the immunoglobulins, eliminate foreign bodies. You know immunoglobulin, very important one, the antibodies are made of this protein. And you know snakes, they have some defensive proteins. Snake venom is a defensive. And some microorganisms, they have toxins, they are also proteins. The test for proteins. Test for proteins means how do you identify proteins in the laboratory. Now, KOH solution, 5% of KOH solution and 1% of copper sulfate are necessary for this test. First you take a protein solution and then add 5% of KOH solution, 2 ml of that may equal volumes of both solution, 2 ml of protein solution and 2 ml of 5% of KOH solution. And then you add 2 to 3 drops of 1% of copper sulfate slowly. Then you get purple color. Now this is the initial solution, and then after adding KOH and the copper sulfate, then this is the color you get. So this is actually confirm the presence of peptide bonds. This test is to confirm the presence of peptide bonds. The last type of organic molecules, nucleic acids. So nucleic acids occur in all living cells because this is very, very important biological molecule in all organisms. They contain carbon, hydrogen, oxygen, nitrogen and phosphorus. 5 elements: carbon, hydrogen, oxygen, nitrogen and phosphorus. So nucleic acids also very large polymers. Very large polymers exist as polynucleotides. Their monomer unit is nucleotide. Monomer unit is nucleotide. So 2 types of nucleic acids can be found in our body or the living organisms. They are the deoxyribonucleic acid, that is the DNA, and the ribonucleic acid, RNA. Now, what is this nucleotide? There are 3 components. There are 3 components in a nucleotide. That is a pentose sugar. Pentose sugar means 5 carbon sugar and nitrogenous base and the phosphoric acid group. These are the 3 major components. So, a nucleotide without the phosphate group is named as nucleoside. Example is adenosine, guanosine. So, phosphate groups in this nucleotides or the nucleic acids gives acidic nature to the nucleic acid because it can donate hydrogen atoms. Now, this is the structure of nucleotide. You can see the pentose sugar, the pink color one. They have this is the first carbon, second carbon, third carbon, fourth one and the fifth one. Right? Now, nitrogenous base is attached to the first carbon. Phosphate group is attached to the fifth carbon. Right? So, this is the phosphate group. You can see the yellow circle. Pink one is the 5 carbon pentose sugar, and this is the nitrogen base. So, this is the simplest form of the nucleotide. You should be able to show this type of nucleotide. Now, this is the pentose sugar and the nitrogenous base and the phosphate group. Now, there are 2 types of pentose sugars. The one is ribose, other one is deoxyribose. Now, look at the structures: ribose, deoxyribose. Now, here all these are same, no? that one oxygen atom. Here the deoxyribose, you can see one oxygen here now is lost. So, this is the deoxyribose and the ribose. Now, in deoxyribose one oxygen atom is less than in ribose, others are same. Now, ribose is present in RNA and the deoxyribose is present in DNA. And again there are two major types of nitrogenous bases, two groups actually. They are the purine bases and the pyrimidine bases. Now, these are the purine structures. Now, you can see they are larger. Larger means that is they have two carbon rings. Each has two carbon rings. Examples are adenine and the guanine. Then the pyrimidine bases. They are smaller because they have only one carbon ring here. So, these are the cytosine, thymine, uracil. You do not need to memorize those structures, but you should know there are two groups of nitrogenous bases as purines and pyrimidines, and what are the examples for each group. Now, I told you monomer of these nucleic acids is the nucleotide. Now, linking of large number of nucleotides by phosphodiester bond form polynucleotide chain like proteins. Linking of amino acids by peptides bond form polypeptide chains, like here linking of millions of nucleotides by phosphodiester bond to form polynucleotide chains. Now, how this bond is formed? Now, look at this. Here the OH group of third carbon combine with the phosphate group and form the phosphodiester bond. OH group of third carbon, there is a third OH group here now, and combine with the OH group of this phosphate group and form the phosphodiester bond. Now, nucleic acids, when we consider a chain means polynucleotide chain, it has this type of nucleotides. Now, you can see nitrogenous base, pentose sugar, phosphate group. 5 to 3 phosphodiester bonds result in a repeating pattern of sugar phosphate unit called the sugar phosphate backbone. Now, these are same in all nucleotides, right. So, therefore, this is named as sugar phosphate backbone. Then these bases will be changed. There are two kinds of nucleic acid depending on the type of the sugar molecules involved. I told you about that. In 1962, these two scientists proposed a special structure or the model for DNA molecule. Actually, they received Nobel Prize for that. Right? So, this is the very important stage, or actually we can say that is the most important invention for development of biology. The scientists are Watson and the Crick. So, they propose the model they propose is named as Watson and Crick model of DNA molecule. Now, let us see what is that. So, they said that DNA molecule consists of two polynucleotide chains. Each they run parallel to each other, and each chain form a right-handed helical spiral. You can see there are two polynucleotide chains. Each chain forms a right-handed helical spiral. Therefore, two chains coil around each other and form a double helix. Double helix. So, it is actually resembles a twisted ladder because nitrogenous bases are appear as crossbars. So, one complete turn consists of 10 pairs, that means this is the one complete turn consists 10 base pairs. Now, there are special features which are identical to this molecule. The one is two chains are antiparallel. Now, let us see what is the meaning of that. The two polynucleotide chains run in opposite direction from each other. Opposite direction from each other, that means the two chains are antiparallel. Look at this. Now, here third carbon here. So, this end is called 3' end, and here fifth carbon, no, the phosphate group is attached to the fifth. This is 5'. Now, this has the 5' to 3'. Here this side has the 3' and this side has the 5'. The chains run opposite direction. That is why we say it is parallel but antiparallel. Parallel but antiparallel. Now, you can see one strand double helix runs from 3' end to 5'. Its complementary strands runs from 5 to 3. Hence, the two strands are parallel but running in opposite direction. That is why we say it is antiparallel. The 5 carbon has 5' end has phosphate group attached to the 5 carbon. The 3 carbon a hydroxyl group here. Here also like that, hydroxyl group phosphate. Then the other feature. Now, you know the base pairing is complementary. What it says? The sugar phosphate backbones are on the outside of the helix. Now, you know that double helix is there. Sugar phosphate backbones are outside of this helix. The nitrogenous bases are inside. Nitrogenous bases are inside. The two strands are held together by hydrogen bonding between nitrogenous bases. But this bonding is specific. That means always purine base linked with pyrimidine base. Purine base linked with pyrimidine base. Now, look at this. The width between two sugar phosphate backbone is equal because of this complementary base pairing. The width between two sugar phosphate backbone is equal as large purine base always pairs with small pyrimidine base. So, in that adenine always bond with thymine by 2 hydrogen bonds, and cytosine always bond with guanine by 3 hydrogen bonds. So, therefore, base pairing is always complementary. Two strands are complementary to each other as base pairing is complementary. Now, remember these two specific features are unique to this molecule. Now, you can see the thymine pairs with adenine. This is larger one with 2 hydrogen bonds, and then cytosine and guanine. Larger one is, this is purine bases, these are pyrimidine. Now, the molecule. Now, this is the 2D molecule actually. In a DNA molecule, when we consider a DNA molecule, number of purine bases are similar to number of pyrimidine bases. Number of purine bases are similar to number of pyrimidine. That means A plus G is similar to C plus T. Purine bases are similar to number of pyrimidine bases. Again, number of adenine similar to number of thymine, and number of guanine bases are similar to number of cytosine. Now, using these things, sometimes you may have some MCQ questions like this one. Now, look at this. In the biochemical analysis of a DNA sample, 30% was guanine. Now, 30% was guanine. What is the percentage of thymine in that sample? What is the percentage of thymine in that sample? I think you can do this. The answer will be 20%. 20%. What are the functions of DNA? DNA is very important because it stores and transmits genetic information from one generation to generation. So, you can see the chromosomes here. Now, chromosomes are made of this DNA and protein. So, according to the base sequence of nitrogenous base sequence of DNA, genetic information is stored. So, that is the very important feature of DNA. So, store the genetic information for protein synthesis also, another function. That means DNA code, that means DNA has the information for all types of proteins. Then let us do the RNA, that is the ribonucleic acids. So, it is a single stranded, not like DNA, and again you know it is differ from DNA by this pentose sugar. It is ribose, not in DNA it is deoxyribose, no. And again it lack thymine. Here uracil is present. That means RNA has adenine, guanine, cytosine and uracil. So, instead of thymine. So, there are 3 types of RNA based on the process of photosynthesis. They are the mRNA, tRNA and rRNA. mRNA is messenger RNA, tRNA is transfer RNA and the rRNA is ribosomal RNA. So, we have to do this in detail in the 7th unit. So, all 3 types are synthesized directly on DNA in the nucleus. All are synthesized on DNA strand. So, let us see the functions of each one. mRNA, it is the least abundant, actually it is about 3% of cell. Functions are copies the genetic information stored in DNA molecule. It copies the information in the DNA molecule. Now, the information actually copied as information copied as a nitrogenous base sequence in a mRNA molecule. Now, DNA has the information as the base sequence. So, it stored that information and transport genetic information from nucleoplasm to the site of protein synthesis. Now, DNA is in nucleus now. So, copying of genetic information occur in nucleus. Then this mRNA carry the genetic information from nucleus to protein synthesis site, that is the ribosomes. And then the tRNA. tRNA is the smallest RNA molecule and linear, sometimes it form a 3-loop structure. Function is transport of amino acids to the site of protein synthesis. Now, when this mRNA come to the ribosome, according to the base sequence, the particular amino acid is transported by tRNA to the ribosome. And the last one is the rRNA. This is the most abundant type, about 80% of the cell contain this rRNA. It has a complex irregular structure. The function is it provides the site where polypeptide chains are assembled. That means the ribosome contains this RNA, that is the rRNA. So, these are the 3 types, and again we can learn about these in detail in unit 7. So, these are the 3 types. Now, you can see the single chain, and this is the rRNA, ribosomal RNA, and this is the transfer. It form a loop-like structure. Other nucleotides. What are the other nucleotides found in living body? Now, look at this. Nucleotides are not only used as building blocks of nucleic acids, but they form several important coenzymes. Now, these are the example: ATP, NAD, NADP, and then the FAD. So, these are the functions. Are ATP act as the universal energy carrier in all living cells. Then NAD, hydrogen carrier, electron carrier, and act as coenzymes. Again, the NAD, NADP function as oxidizing agents in respiration and photosynthesis respectively. FAD, the other one, act as coenzyme or act as electron carrier. Now, I finished the chemical basis of biology. So, today, now I hope to wind up the lesson. Have a good day.